Okay. It is my distinct pleasure to introduce the moderator of today's webinar, Bert-Jan Korte, who is currently a PhD candidate at the Hubrecht Institute in Dr. Alexander van Oudenaarden's lab, where he focuses on developmental biology and stem cell research. He completed a master's in molecular and cellular life sciences at Utrecht University under the supervision of Dr. Francesca Mortiroli, where he also gained teaching experience as a student teaching assistant. And his strength in research, writing, and communication makes him a valuable contributor to the scientific community. Thank you so much for being here, Bert. And
thank you so much.
Hand it over to you.
Okay, thank you. So I'm here to introduce the speaker of today to you, which is Jeroen van der Berg. Jeroen completed his PhD in the lab of René Medema here in the Netherlands at the Netherlands Cancer Institute. Here he worked at investigating the relationship between DNA damage-induced cell fate decisions and the genomic location of DNA lesions. After his PhD, Jeroen started working in our lab, the Van Oudenaarde lab, at the Hubrecht Institute here in the Netherlands, where he has focused on developing techniques for studying replication dynamics in single cells. This has resulted in the development of single scEdU-seq, which is a technique that he will be presenting to you today. And so with this, I would like to give the floor to Jeroen. Take it away.
Thank you very much, both of you. Okay, yeah, it's my distinct pleasure, and thanks to the Bioprotocols community for the invitation to allow us to present our method, single scEdU-seq. Today, I'm going to go through some of the aspects that we can that we can quantify using single scEdU-seq, and give you some examples, which to us are really interesting in quantifying some of these DNA replication dynamics in single cells. But first, I'll start off with my introduction slide. Yes, there we go. So DNA replication is a complex process which is required to copy the entire genome of the cell. These DNA, you see the machinery responsible for this DNA replication process here, which I'll refer to as a replication fork or a replosome interchangeably. And these processes in genome start at places of DNA replication origin, where one of these machines is loaded, where one of these replication origin machines is loaded, and then from that, this origin fires, so to say, and then two replisomes are born that start to replicate the genome in opposite directions. However, if you would only have a single DNA replication origin in a cell, you would one cycle of DNA replication in human cells would take over 1,000 days. So what the cell has done to accommodate that is to have thousands of these active DNA replication forks at any given time during S phase, and these are all copying the genome at a steady rate. So why do I think working on DNA replication is important? So, and especially these dynamics. So you can imagine that once one of these sort of replisomes or replication fork machines encounters an obstacle in the DNA, that these obstacles, such as DNA damage, need to be repaired before the replication machinery moves over, and an error is made that cannot be reverted anymore. So intrinsically, these DNA replication, these obstacles are affecting the rate at which these DNA replication machines can progress over the genome. We know from work from many different labs that if you do not properly handle these obstacles, you get massive genome instability, as depicted here with these lines being translocations between different chromosomes, these green and red bars indicating amplifications and deletions. And initially, a lot of this work on DNA replication dynamics have been done by a single molecule assay, which is called DNA fiber assay, by which they can extract many of these replication dynamics from a population of cells. However, these DNA replication dynamics might depend on the underlying DNA sequence, chromatin, cell state, or cell type, with which the fiber assay is largely agnostic. So using a fiber assay will not answer most of these questions. So in order to address that, we wanted to develop a method with which we could probe all these different things in combination with these DNA replication dynamics. So I'll shortly start with a workflow, and then I'll keep on going to the rest of more of the analysis. So what we do for most of these single-cell EDU-seq workflows is that we sort S-phase cells, and this is a representation of Fuji space, which is a cell cycle fluorescent probe systems. And with this, you can sort S-phase cells, but this works just as well if you would use a DAPI label for cell cycle progression. We then go to a flow cytometer, and with this flow cytometer, we extract S-phase cells either by this Fuji or DAPI, and then sort them in 384 well plates. And then with these 384 well plates, we can then perform our molecular biology to extract the relevant DNA replication tracks. So what we do is that first we start with incubating the cells with EDU, then we get a labeled stretch of DNA, here indicated in blue. And we can do this for a variety of labeling times. And once we fix the cells, we can use click chemistry to click on a biotin on this EDU molecule. So now we have specifically clicked the biotin molecule on our EDU labeled DNA. Subsequently, in these plates, we now digest the genome with a restriction enzyme. And after this restriction enzyme digest, we ligate adapters containing a cell barcode to refer back to which well this particular cell was sorted into, and a unique molecular identifier, which allows us to find unique reads in these single cells. After that, we take all the reaction volumes from these 384 wells, we put them together, and then we perform a magnetic bead separation with streptavidin. And as you might know, streptavidin binds to biotin. So now we specifically pull out only the pieces of the genome that have been replicated once we've added EDU. Then we heat denature our DNA from these beads, and then we get the complementary DNA strand of the biotin-related DNA. This we prepare and then subject to DNA sequencing. So in order to show that this works, we initially started to validate our method by comparing it to an already existing method, which is called replication. So here you can see a region of chromosome 1 in the genome where we have taken cells that are early replicating and cells that are late replicating, which is the staple of this DNA replication method called replication. And you basically look at the regions in the genome which are either early replicated, indicating by a signal that is positive, or late replicating, that is signal that is below 0 over here. And what we can see here is that our single-cell EDU-seq signal nicely behaves behaves similarly as the replication signal. And this is, of course, a small part of chromosome 1, but this also maintains for all the genomic locations throughout the entire genome. So, and where does this, now we have some single cell resolution, right? So now we can ask, where does our single cell EDU-seq signal come from? So what we can show over here is our cells that pass our QC, or we call them single cell EDU-seq positive cells. And then, so these are basically cells that we select over here in this quadrant between the dashed lines, we consider to be EDU-seq positive cells. So we get a certain number of reads from these cells, and this signal is not behaves as we would expect from single cell sequencing data. If we then look at where these cells, because we sort these cells and we get information from the flow cytometer, we can now ask, where specifically do these EDU-seq positive cells fall in a DNA content plot. And that would be something like this. So here you see that now I've labeled the cells here that are here in gray and blue, but then on the x-axis you see the amount of DAPI signal that we find in these cells, and you see that there is indeed it does not fall. So the EDU-seq positive cells does not follow the same distribution as the EDU-seq negative. So in the EDU-seq negative, you see this large G1 peak where there's like cells that have not yet started DNA replication. This does not occur in the EDU-seq positive cells. So there's definitely a selection for these cells. And you see that you get an almost equal amount of EDU-seq positive cells over the DNA content. At a certain point in time, this drops off as the cells enter into G2, which is the next phase of the cell cycle. And you see that again, the EDU-seq positive cells do not follow the similar distribution of the DAPI content, which we see in the EDU-seq negative cells. So this is all very nice, but now we wanted to order these cells to sort of figure out how do we now, how does DNA replication behave as a function of time? So in order to do that, we decided to not use this DAPI intensity, but we decided to use the signal we have in our single-cell EDU-seq assay. And for this, we made one assumption, and the assumption was that once DNA replication occurs, it occurs linearly on the DNA. So that means that once an origin is firing, DNA replication machine moves from point A to point B in a linear fashion. That means that if you look at it in a genome, on a genome, is that a replication machine that is a little bit further in S phase will have progressed a little bit further. And this is what I try to illustrate in this particular plot, where we have three different replication tracks, so sizes of positions of EDU incorporation on a fictitious chromosome, and then we basically use algorithms to tell us which cell is now most likely which is the closest to each other. So as you can see over here, we can now fit a distribution, which we picked the Gaussian distribution in this particular case, and then we overlaid them on the center of this blue and red. And then what we can do is then we can ask, what is the overlap between this signal? So the overlap between the signal obviously is the largest, or at least the closest, is blue is closest to red. So, and this is what you see over here. You get this overlap score. And by taking this overlap score and then using a UMAP-like algorithm, we can now ask which cells are most likely closest to each other, and can we then compute an S-phase progression from this? So does the EDU-seq data give us something that is actually telling? In order to validate this, I need to explain the Fuji system a little bit better. So the Fuji system is a ubiquitination-dependent cell cycle indicator, which is tagged with fluorescent probes. And you can basically see it as a circular progression, as you can see over here. So cells in early G1 will become increasingly more red until they enter into S phase, and then they will start to create the green fluorescence. So as an overlay, that will be yellow. And then it will lose the red signal, and then it will accumulate the green signal until it enters into mitosis and then continue on. So basically, you can read this as sort of a circle basically around this axis. So in order to sort of show that our S-phase progression that we've calculated over here, which seems kind of abstract, does fit to the data, is that we've sort of taken this S-phase progression score, which we've put between 0 and 1, and then we've looked at what are now the fluorescent intensities of these fluorescent markers, these Fuji markers. So indeed, what we see is that cells that are early in S phase are green, are red, as we see over here. And then this red decreases as a function of the cell cycle, which we also observe over here. And in the green signal seems to accumulate as the cells progress further into the cell cycle. And maybe even more importantly here, the DAPI signal, so the DNA content, also positively correlates to the progression throughout S phase. So cells start their DNA replication journey over here, and then they increase their DNA content as they progress through the cell cycle, progress through DNA replication. So what can we now do with this? So with that, we can generate images such as this, and this is, I think, a lot to put on the screen in once. So I will take my time. So here you can see a heat map of a DNA replication progression over S phase. So here you have 30 megabases of chromosome 2 on the x-axis. And on the y-axis here, you have this S phase progression where the 0 is the start of DNA replication and 1 is the end of DNA replication. What we can observe in these kind of plots is that there is a highly structured signal. Most of the signal is most of the plot is mostly empty. So there's like there's not DNA replication everywhere at any given time, but it seems to be highly structured. And for instance, to give an example, we see this one single lonely DNA replication fork, which exists for a very long time in S phase. Then we have taken our fluorescent data again. So here we have the Fuji red, which decreases over time, the Fuji green, which increases over time, and then the DAPI signal signifying DNA content that also increases as a function of this S phase progression. To show that this is now something that makes sense, there was data that was generated both by the ENCODE consortium, which is the top bar over here, which signifies replication timing, which is what I also discussed before, this early versus late DNA replication. And what you see is that indeed these red areas over here correlate nicely to regions that are early in DNA replication, and these blue stretches are very well correlated to these regions that are late in replication. Another method that another lab, that the Hallucinati's lab has pioneered, is called EDU-seq H2. And what they do is that they add a very high concentration of a DNA replication inhibitor to synchronized cells. And what you then can see is that you only get the very first part of DNA replication. So the first 2 or 3% of DNA replication. And what you can see is that these peaks, as you might call them, align very well to these early DNA replication regions that we identify with our single-cell EDU-seq. I mean, this is, of course, looks, I mean, I think this looks very nice, but can we now also extract some quantitative aspects from these type of plots? And in order to do that, we can now, what we do is that we want to sort of estimate how many of these blue stretches there are in a single cell at any given moment. So we use a hidden Markov model, and with this hidden Markov model, we can now detect the number of DNA replication forks in single cells. And what we can see is that indeed, this is a quite stable number over this S phase progression. So throughout DNA replication, we see that there is a given number of DNA replication forks that follow almost a Gaussian distribution. And this is a very nice metric for us, because now we can use this to sort of ask ourselves how sensitive is our method. So in order to do that, we can downsample. So this is something that's done often in single-cell RNA sequencing. We can downsample the number of unique reads. So here at 100 would be all the unique reads you had for a single cell. if you now say, I will take a quarter of my unique reads away from this data, how many of these DNA replication forks do I still detect in a single cell? And what you see is that, that we can downsample quite far for most of the cells before we actually see a decrease in detection of these number of DNA replication forks. Well, this to us indicates that it is nice to have these many reads that are unique, but it seems that we do not need all of these reads to make an accurate estimation for DNA replication speed forks. Okay, so now I'm going to shift gears a little bit and stop talking about the number of forks. I'm going to start talking about speeds, because this was in the title, or at least implied in the title of the manuscript. So what we do to detect DNA replication speeds is that we use a pulse chase label experiment. So that basically means that we subject cells to 2 pulses of EDU. And in between these 2 pulses of EDU, we leave a gap in which we do not label. So then we get in a linear genome, we get 2 patches of EDU positive DNA. As I said before, we can extract this particular EDU genomes. We can then do single cell DNA sequencing, as I also described before, and then we can quantify the speeds, in which I will come back to later how we do this. But also what I told you before is that we can do DNA replication timing, fork number, etc. So basically, how do we now, from these 2 pulses, quantify our DNA replication speeds? Because a logical thing to do would be to measure the distance between these 2 pulses and divide it by the time. And that is more or less what we do. So first, I want to explain to you why we chose to do a double pulse experiment and not a single pulse experiment. So you can imagine that if you have a single pulse experiment, you can actually measure the width of, so these now signify reads, single reads. So you can basically, for a single pulse, you can now measure the width, and the width would be the speed at which this fork would progress through the genome. However, if you would miss one read because you haven't sequenced deep enough, or there's some problem with the experiment and you haven't sort of extracted all the information you needed, you can lose one read. And if this read would be at the end of the, so this is also, I guess, worst case scenario, if this read would be at the edge of this this replication fork, then you would say that this speed would be 30%, almost a little bit more than 30% lower than it would in reality be. Now, if we would use a double pulse speed measuring exercise, we can basically lose one read from each of the pulses, right? So in that sense, a similar amount of reads per pulse, but still our speed estimation would be almost 100%. And I guess this is illustrated in this simulation, where we have taken on average 2.5 reads, 3.5 reads per one of these tracks, and then sort of remeasured, if we then remeasured what the speeds are in these particular conditions. And I think sort of the take-home message for us here was that if you now get into this lower regime of number of reads per track, you basically start to largely underestimate the DNA replication speeds in these conditions. So it's here signified as very small points, but here in the bottom it's box plots, so maybe a little bit easier to interpret, is that you see that if you now have low number of reads per one of these replication tracks, you start to underestimate these DNA replication speeds in a single pulse experiment, but you do not seem to do this at all if you have a double pulse experiment, basically. So this gave us, this exercise sort of told us that it was probably the best way to measure DNA replication speed using a double pulsing strategy. So how does that now work more technically? So if you envision it, it's all about the distances. So how far is stuff away from each other? So if we would have, let's say, a number of forks here that would have progressed, that you would have labeled twice. So let's take this example for one. You have a variety of different distances between one of these pulses, which are here indicated in yellow. And these also exists for these other ones here for the second pulse of this particular fork. But of course, you also have the distances that span these two pulses, and that these are these cyan colored distances. This happens, of course, for many different forks. And now, if you have many different forks, you of course get also distances between all the forks, which makes it a little bit complicated. But I think we were sort of lucky enough that sort of with a correct labeling scheme where we can sort of control the time of the experiment, we sort of know, we sort of have an idea of where this particular these cyan reads should pop up versus the purple reads. So basically, we thought that the distribution of these distances, so if we now sort of take only the distances and then ask how frequent are these distances occurring in these single cells, we can basically build up a sort of histogram of distances, so to say. So if you now, so these yellow reads, which would be within one of these pulses, would be distributed as an exponent decreasing very rapidly because they only label reads within one of these pulses. Then you would have a lot of these distances that are sort of interfork long-range distances, which we assumed would form a uniform background. And of course, we get our cyan distances, which is what it's all about, right? So we want to sort of measure the distances here. And then our idea was if these form a nice distribution over all the forks, we can now sort of extract this cyan distribution and then measure the average of that distribution. And by measuring the average of that distance, we can now divide that by the time it took to between these two labelings of these pulses. And if we divide these two things, we get our speed estimate. So this is all nice theoretical, but doesn't really contain any data. So what does the data now look like? So the data actually does behave quite as you would expect. So here you see a single pulse experiment. So this would only be the yellow distributions, right? So we don't have any cyans here as far as we know of. So what we see is that we get a very nice exponential decrease of this histogram that all go down and then basically remain flat in this uniform purple background I was telling you about. If you now do a double pulse labeling with different times in between these double pulses, you start to see that there's indeed an extra sort of hump coming into the data. And this at 45 minutes doesn't seem sort of well defined enough to really sort of extract this. However, if we now do this with, it's called 75, but it's 60 minutes because it's distance between the middle of the labeling, so it's 75 instead of 60. But basically, you now start to see a very defined sort of hump in the histogram of distances. And similarly, if we now sort of increase the time to even longer, we see that this is now shifting even further to the right, indicating that we're sort of capturing these forks in the process of their copying, in their copying process. And what we think is very nice is that this is sort of highlights the sort of single cell aspect of this method, because if we do this with, in this case, 5-cell bulk, so we take all the signal from 5 cells, merge it, we basically get a flat stripe, meaning that, you know, we can only do this measurement in a single cell, which I think is very nice because it now allows us to, within a single cell, really measure this distance distribution. On the other hand, it's also very nice that this 5-cell bulk doesn't show any signal, because that would mean that we're measuring something else, because the signal would be not necessarily all at the same place. So this should this behaves exactly as we expected. And moreover, if we now take this one cell again and then downsample it, so what I was saying before again, it's like taking, instead of 100% of the unique reads, taking 50% or even 25% of the unique reads, we still see this same distribution in our data, indicating that, you know, there's quite a lot of signal that contributes to these to our ability to measure this second hump, so to say. So this is all very technical, but what does it now mean for, like, is there any real biology in these data? So what we did is subject regular non-transformed RP1 cells to this method. And then again, here I show this S-phase progression. So this alignment of progression through S-phase from the start to the end of DNA replication, and our measured DNA replication speed, as I just explained to you, this middle of this cyan distribution. And what we see is that these DNA replication speeds are not constant as a function of this S-phase progression. So we see that regions that are replicated early in S-phase seem to be much slower than regions that are replicated towards the end of S-phase. And this we could also validate using a classical fiber assay. We had to tweak the assay a little bit to do that, but what we did is that we sorted early S-phase cells with this Fuji marker again, and late S-phase cells. And now we quantified the length of this labeled nucleotide track, which is a metric for DNA replication speeds. And indeed, what we found is that cells that are in early S-phase have shorter track lengths or fiber lengths than cells that are in late S-phase. So this was very nice for us to see that this was indeed something that was happening, also based on the assay that was most widely used in the field. So, but what might now explain this particular acceleration or increase in DNA replication speeds? So what we found initially is that if we overlay this with single-cell RNA sequencing data, we found that early forks, so these DNA replication forks that are transcribing these early regions in the genome, are replicating transcribed genes. So actively transcribed genes are preferentially replicated in early S-phase, and that the earliest DNA replication forks encounter the highest level of transcription. So although it's a lowest fraction here in dark red, it seems that if you look at the normalized coverage of EDU-seq over this S-phase progression, it more often encounters a highly expressed gene or high to mid-high than it encounters a lower medium expressed gene. So it seems that regions that are early replicated in genome, regions that are early replicated in genome are covered by transcribed regions, or are also transcribed, I should say, basically that. So next we wondered, so how is transcription now actually responsible for this decrease in or these lower replication speeds? In order to do that, we used a RNA polymerase II inhibitor, which is called DRB, where I've labeled now the single cells here in or the cells in cyan for the DRB condition and gray for the DMSO. What you see is that mainly in early S-phase, we see an increase in DNA replication speeds if we treat cells with a transcriptional inhibitor. And this is another way to quantify this, where we look at the differences in speeds over DNA replication between the two conditions. And then we see that due to the RNA polymerase II inhibitor, we approximately get 400 base pairs per minute faster DNA replication in early regions or in transcribed regions, because we know these overlap. But in order to formally quantify that, we can now quantify the DNA replication speeds inside, so an orange inside transcripts or outside transcripts, what we see is that there is indeed, although a little bit less, there is a slower DNA replication inside transcripts than outside transcripts. But if you now treat these cells with DRB, so this RNA polymerase II inhibitor, we see that this effect is completely lost. So, and next due to my background working in DNA damage as a PhD student, I wondered whether or not this has something to do with the vast amount of literature that people have previously described that transcription and DNA replication form sometimes enemies on the same template. So what we did is on a flow cytometry assay, ask what now happens with these two DNA damage markers if we treat cells with an RNA polymerase II inhibitor. And what we observed is that both gamma H2X, which is a phosphorylated histone variant, decreases in the DRB condition, so here in cyan, and ADP ribose as a readout for PARP activity also decreases. So does that make sense? Well, there is, as I said already, quite extensively reported interplay between transcription and DNA damage. There's multiple different things which I don't have time to go overall, but you can think of it as RNA polymerases being a problem to DNA replication as creating supercoils, which then need to be resolved by topoisomerases. There is a high turnover of DNA methylation in promoter regions or around promoter regions and in transcribed regions, which sometimes require base excision repair to remove these unfortunate lesions. And somewhat worst case scenarios, you can also get topoisomerase II activity that really breaks the DNA apart. And this now also creates an obstacle to DNA replication, but maybe more described in literature are these R loops, which is an RNA-DNA hybrid. So this is when an RNA molecule hybridizes with a DNA strand instead of its complementary DNA strands. And these exist when, these can be generated once there are once DNA replication and transcription inhabit the same strands. For instance, if RNA polymerases are in the process of transcribing, but then halted, then the nascent RNA can now form a DNA-RNA hybrid, which forms, which is recognized by DNA damage signaling, and can now form a problem to the oncoming DNA replication machinery. So this is, I guess, what was known. So what we did to prove that interaction was to inhibit DNA damage signaling and mainly PARP signaling. So what we did was inhibit PARP inhibitor, or what was inhibit PARP by olaparib, a PARP inhibitor. And what we see is mainly, we see very comparable pictures what we saw with the DRB experiment. So we see that DNA replication speeds in PARP inhibitor treated cells are higher than in DMSO, but it seems to be mostly the case in early regions, so these regions that are transcribed. So in order to test that the DNA damage restricts DNA replication speeds not only in this particular condition, we wanted to activate PARP enzymes, and we did that by using a genetic knockout. So normally PARP enzymes are recruited to sites of DNA damage, and then via its auto PARylation, so it modifies itself with ADP ribose, it can recruit this DNA repair complex, and this DNA repair complex then comes in to repair repair this lesion, and then it can move from this location. However, if the XRCC1 is not present, this PARP enzyme starts to hyperactivate and starts to modify itself even further. So in order to sort of address whether or not PARP activity is sufficient to decrease DNA replication speeds, we used this knockout in which we clearly see that DNA replication speeds are lower in this XRCC1 knockout compared to our wild type cells, and this you can also see now in this difference. So it seems that there are like 200 base pairs to even 400 base pair per minute slower in this XRCC1 knockout. So now I've talked about speeds and how we can quantify this and uncover some interesting interactions between transcription, DNA damage signaling, etc. However, there's also another measurement we can make from this distribution that I haven't told you about. So again, what we've done up till now is only quantify this midpoint of this cyan distribution. But of course, the distribution is defined by more than just the mean. So we can also quantify the variance of this distribution. So basically, how wide is this? And how we interpret the width of this distribution is how similar are all DNA replication forks progressing in a single cell. So we took all our data that I've shown you up till now, and we can now basically make a plot in which we can now show DNA replication speeds on the x-axis. So you go from low speeds to high speeds, and then a measure for the standard. So we quantify the standard deviation as a sort of representation of the variance of the signal. And then you see that indeed there's quite some conditions that physically separate from other conditions. And this may be more appreciated if you put them in separate graphs, where you can see that if we if we have wild type cells, we get a high density of speeds around this particular condition, and then a plume of irregularly forks at irregular speeds over there. If we then treat these cells with DRB, we see that mainly the variance seems to decrease as well as the speed seems to increase. So it seems that these forks are no longer moving, are now moving at the same speed, and they're faster. And if we use a PARP inhibitor, we see that they're even faster, but they seem to be a little bit less synchronous, maybe due to the fact that they encounter lesions that they are physically stopped by, that are now no longer sensed in the PARP inhibitor condition. And again, here we also see the same if we use this XRCC1 knockout cells that have elevated PARP signaling. And indeed, we can also repress this variance increase by treating these cells with a PARP inhibitor. So now I want to this is all sort of part of the initial story. More recently, we've collaborated with a lab in our institute that are interested in de novo histone deposition. So this is a process that occurs simultaneously during DNA replication. And one of the critical factors and players there is the CAF1 complex. What our collaborators have done is to use a degron to very rapidly degrade this complex so that there's no longer any new histones. So these new histones are then no longer inserted as DNA replication passes through. So what we found using their cell lines is that there is a very rapidly, so I need to explain this a little bit better, but so we use DMSO cells, and indeed, again, we see this increase in DNA replication speeds in our control-treated conditions. If we treat these cells with DTAG, so if we get rid of this CAF1 complex, we see that they start out at more or less similar DNA replication speeds in the beginning of S phase, and then they seem to decrease as a function of S phase, but cumulatively. So they seem to be getting slower and slower and slower. And interestingly, I think much in line with our previous data on the variance, we see that the heterogeneity, so this standard deviation of the fork speeds in these single cells, seem to increase as a function once we get rid of this CAF1 protein complex. And furthermore, we can now also see that indeed this heterogeneity or standard deviation measurement of these DNA replication speeds seems to be mainly affected later in S phase, where we also see the largest decrease in DNA replication speeds. So to sort of wrap up, I guess what we're interested in and what we sort of tried to describe in our initial publication was using single-cell EDU-seq was how do RNA polymerase do transcription, how does this now sort of affect obstacles, so create DNA damage, which recruits these PARP enzymes, which now sort of represses the sort of DNA replication machinery to progress. So sort of our own perspectives, or what we want to do in the past, is sort of really dive deeper into what sort of dictates this increase in DNA replication speeds over this S phase. So you could interpret this as what is the question mark here, but also a very interesting observation is that there's also quite a bit of variability of DNA replication speeds between cells that seem to be replicating the same stretch of genome. So even though the trend seems to suggest that the time in S phase predicts how well, so what kind of speeds you have, we also see that there's within the same, stretch of cells, there seems to be quite a big difference in DNA replication speeds. So that's where I want to leave it off. I want to acknowledge a few people, mainly Alexander for having me in the lab and supporting me throughout this entire endeavor. Vincent, who is a very talented PhD student who helped with a lot of the analyses for the initial publication. Bert-Jan for being a moderator here, but also joining the team later on and addressing a couple of the questions that I stated later at the end of my talk. Our facilities, both sequencing and flow cytometry, our collaborators in Denmark, with which we recently got a grant. Our other collaborators, mainly Francesca Mattioli, from which I loaned the CAF1 data. And if you are interested in this type of research, we have on the collaborative grant with the Growth Lab, we have open positions for postdocs and PhD students. You can email me if you're interested. And I would like to thank you for your attention and open for questions.
Thank you for the talk, Jeroen. So there have already been some questions coming in from the audience, so I'll start with that. Specifically, Tatiana asks, what percentage of folks travel uninterrupted for 75 minutes? And she or he has simultaneously some follow-up questions, mostly for forks where the origins are closer, wouldn't they complete replication within the 75-minute time frame? And in these cases, how do you deal with this? Do you discard these areas where these origins are closer?
Yeah. Yes, there are definitely forks that annihilate each other as well as or terminate into each other, and forks that are born through origin firing. So based on data that I didn't have time to show, we estimate that we measure approximately 40% of the DNA replication forks we can assign from 40% of the replication forks we can extract these DNA replication speeds. in early S phase, it is indeed an issue because they're especially the initiation sites within a single initiation zones are much closer packed than our 75-minute measurements, because these are described in literature to be 30 to 50, well, somewhere 30 kb away. Yeah, these we would completely miss. However, we've done some simulations to show if the spacing that we and others have observed for origins would affect or would create an artificial DNA replication speed, and that we do not find. So it basically comes down to the fact that DNA replication origins are not interspersed at a regular enough interval to interfere with the measurements that we do on fork speeds. And I hope that answers your question.
Okay. The second question coming in from Angelo, who asks, can single-cell EDU-seq be used to discriminate reads coming from leading or lagging strand synthesis?
Yeah, that would be great, wouldn't it? No, as of yet, it doesn't. So we are mainly measuring. So we're measuring a convolution of both leading and lagging strand synthesis. We are in the process of thinking, especially now that we have a strong collaboration with the Growth Lab, to make this method stranded, similar to their scEdU-seq protocols, and then we would be able to measure that. One caveat, though, would be that we are, for most forks, we retrieve somewhere between 5 and 10, if we do single pulse, that is 5 or 10 reads. I think it's a little bit on the lower end of what we would be comfortable with, because we need to divide that number by 2. So then we have 2 or 3 reads on the leading and lagging strand. So we need to think about our labeling strategy as well a little bit. But yeah, it's definitely an option, and I would really love to sort of think about that. Yeah.
Okay, great. So there's a question I've been reading a lot, especially on the website, but also here just now in the chat, which is how does single-cell EDU-seq compare to the standardized technique of DNA fiber analysis? And are there positive, negatives, or big differences between these techniques?
Well, yeah. So I think for measuring, so there's a couple of things you can't measure with EDU-seq. There's a couple of things you can't measure with a fiber. So I think in that sense, they're complementary. As for instance, measuring I think origins and exact placements of origins are quite difficult for us. Our lowest resolution we have is 20 kb. If we push it, maybe 15 kb. So any and all sort of measurements, as I was just describing, with multiple initiation sites in an initiation zone that's described to be 30 kb, we are completely blind to that measurement. And I think in terms of DNA replication speeds, I think we get for most of the cell lines that we have processed now in ongoing collaborations, we see extremely similar DNA replication speeds. So I think it is quite interchangeable on that aspect. However, using a fiber assay, it would be, I think, quite difficult to quantify DNA replication speeds in certain S-phase timings and in certain places of the genome, which we can do with EDU-seq. And one thing that the bulk single molecule, but still bulk nature of the fiber assay does not allow for is to quantify the number of DNA replication forks in a single cell, which doesn't seem to be that altered in our control conditions that I showed previously. But we are now sort of gathering more data, and we see that there's quite some heterogeneity in the number of forks. And this now also affects DNA replication speeds in the same cell. So I think there's quite some underappreciated complexity in the number or in single cell assays in that sense that might be lost in the average of the fiber assay. Yeah.
Okay, thank you. We also have a question by Susanna who asks, do you think that the changes in speed are related to regions that are difficult to replicate because of potentially their repetitive nature? For example, are centrosomes and telomeres replicating late in these cells?
Yes, they are. And no, I don't know. So that's the honest answer, I think. So both centromeres and telomeres or subtelomeric regions are replicating late, at least in these RPE cells. We haven't. So the issue there is that the repetitive nature also makes it quite difficult to make these estimations. And we haven't mapped these data to a telomere to telomere genome that would be necessary to make these analyses. But in principle, if regions of the genome would be unique enough, we would be able to extract DNA replication speeds from them. Yeah.
Okay. There's another question by Salah who asks, how does EDU incorporation affect the fidelity of DNA replication? And how do you control for any potential artifacts introduced by EDU during the sequencing process?
Yeah. It's a very good question. So I think the concentration of EDU that we use in these particular experiments are concentrations that a lot of labs have used in the past, and also we've looked at whether or not we can see any defects in cell cycle timing with our concentrations of EDU, and we do not observe any delay in cell cycle progression based on the concentration of EDU. So there's no overt sort of toxicity or whatever related to the EDU. And with regards to the artifact of the sequencing, I mean, in principle, we're not sequencing the EDU molecule, right? So we're sequencing the complementary strand of the EDU molecule. But I mean, this might indeed have some unforeseen effects on our library prep that we're not aware of. That's, I guess, why we always try to do multiple biological replicates in these experiments to at least sort of get that out of the system. But I mean, they might.
Okay. So another question, which is more a bit technical. Priscilla asked, what is the sequencing depth required for single-cell EDU-seq? And maybe we can combine this with a question from the website, which asks, how many cells do you normally need to apply this technique?
Yeah. So to start off with the first, this is something we're extremely happy with. So thank you for the question. So we need roughly somewhere between 20 and 30,000 unique reads per cell. If you compare that to, for instance, single-cell repli-seq, which, the numbers are different between publications, but I think there they often suggest to go more than 1 million reads per cell. So that's 40x more in that sense. And that is due to the fact that we particularly purify only DNA replication molecules from a pool of many different molecules, and then we can only sequence those particular molecules. So relatively little. In terms of single cell sequencing and depth and how much we need, it's actually quite economical in that sense. In terms of how many cells do we need, I guess the boring answer is it depends on your experiment, but the realistic experiment is that we generally aim for, so we do it in a 384 well format plates. So normally we process somewhere between, we either do 3 or 4 plates per experiment, but we've gone lower for other experiments where we've seen that 2 plates is also enough for an experiment. So then you would be talking about 780 cells, 760 cells, sorry, per experimental condition.
Okay. There's a question by Salah who asks, how do you envision the application of single-cell EDU-seq in studying diseases related to DNA replication stress or dysfunction? Are there any particular diseases or conditions where this method could provide novel insights?
There's a lot of DNA replication, or there's a lot of syndromes in which DNA replication proteins are affected by point mutations. And interestingly, there's a couple of these that have very tissue-specific defects. So I think there's some of the polymerase epsilon accessory factors that have been mutated. They are mutated in certain syndromes, and these seem to be particularly affected in the lymphoid compartment, but not necessarily, and this is of course highly proliferative, but not necessarily in the intestinal tract, which also has to deal with a lot of proliferative activity. So I think in that sense, so talking about genetic disorders, there's a lot of stuff that we do not really understand how a single point mutation would create differences in sensitivities or toxicities in different tissues. And on the other hand, and this is maybe something for more of the future, but we're trying to think about, integrating EDU-seq with a variety of other measurements, such as transcription. And we know, of course, that transcription and replication conflicts pose a big problem to the genome. So in terms of, recently also a paper about transcription replication coupled mutational signatures, and how these are found in cancer and how these are sort of predicting overall outcome. However, we do not really know. I think we have been somewhat unable to measure sort of the distances between these processes because we've been measuring averages basically, and since the position of every RNA polymerase or DNA polymerase can alter slightly between each individual cell, it will be very difficult to sort of ask the question is so how close do these two machines need to come together to form a real problem to the cell to really create DNA damage? So I think that's something that we're also quite interested in, especially based on the data that we've already published on this with regards to transcription replication conflicts, PARP, and that.
So during the talk, there's also a clarification question from Charlie who asks, how do the PARP inhibitors speed up DNA replication while they themselves cause DNA damage?
Yeah, that's a good question. So I think we still do not really understand. I think we still do not really fully understand how this works. So I think the lab of Thanos Halazanaitis also last year had a paper about how, for instance, transcription inhibition can alter PARP inhibitor sensitivity, even in BRCA negative settings. So that seems that there is definitely some overlap. There's also been quite a lot of mass spec studies performed mainly, many different labs, but for instance, the Lund Nielsen lab has done a lot of mass specs on this. And there's also a lot of physical targets of PARP in the sense that a lot of proteins are also ADP ribosylated. And this is also, if you look at this list, it's both transcription and DNA replication. So it might be that, it's not only sensing the damage and creating the damage as a PARP inhibitor, but it's also robbing the cell of the ability to adapt to the damage, basically. I think that's probably what's happening, but I mean, I think we need more data to prove that, but I think there's probably more happening than just DNA damage with PARP inhibitors.
Okay. We have another question, which is partially also from the website, which asks, can single-cell EDU-seq and/or these DNA fiber assays be used to measure replication dynamics in more bacterial/prokaryotic systems? Is this something you have looked at?
No, we have not. And yes, in principle. So I know, for instance, in yeast, you need to use specific yeast strains to incorporate BRDU, because BRDU is not normally incorporated in yeast cells. So they need a mutant line, which I think expresses a human protein to make the BRDU incorporate in the genome. And so I don't know if that would also be the case in the bacterial conditions, but it's likely that it wouldn't work immediately out of the box. But yeah, I mean, in principle, if it incorporates, and we could in principle do these assays in any species.
Okay. I think there's still one question which I have not answered, which is from Akita. I'm sorry if I am saying the question wrong, but I interpret it this way, which is, do R loops in cells increase DNA damage? And have you investigated how this affects the replication dynamics of these cells?
Yeah, that sort of comes to the fact, are secondary structures DNA damage or not? So I think there's quite some evidence now to suggest that non-B-form DNA, so secondary structures like R loops, but also G4 quadruplexes, hairpin structures in the DNA, are recognized as DNA damage, and there's more and more papers coming out, I think, every month. And interestingly, I think there is a paper, and now I forget from which lab, for which I apologize, which describes that R loops also recruit PARP enzymes, which then activate and then basically recruit all kinds of RNA helicases to resolve the R loop. So I mean, it is recognized as a form of DNA damage. So increased R loops does associate with higher levels of gamma H2X. It does increase ADP ribosylation by PARP. So the cell sure thinks it's DNA damage, I think. I think that's the correct answer.
Okay. I still have one more practical question, which is more from the side, which asks, what are the main challenges faced when implementing single-cell EDU-seq in a laboratory setting?
Yeah, so I think the real challenge lies in low volume pipetting robots, right? So I think these do exist in many university labs, institutes, etc. So our dispensions, or low volume dispensions, so that's the dispensions of the restriction enzymes or the DNA ligase reactions that I described in the beginning of the presentation. These range between 50 and 150 nanoliters. So those are quite small volumes, but these are achievable by using robots. Yeah, and I think the entire most of these protocols, these plate-based single-cell protocols, fall and stand by the access to one of these machines. But there are quite in my experience, these do exist in the wild. So I think if you would want to, then and often they're in like chemistry departments, a lot of what do you call them? X-ray crystallographers use a lot of these machines to do dispensions. So yeah, these do exist, but I think this is the biggest bottleneck, is that the robotics, the low volume robots are low volume pipetting robots are the biggest hurdle to overcome. The rest is quite doable, I think.
Okay, these were kind of the questions that I mostly see pop out. If there's still people in the chat who want to ask a question, maybe we give them a few seconds to still ask the last ones. But other than that, I think it's been very nice. Let's see.
Hey, well, there's a lot of thank yous coming in. Thank you both for a really interesting talk, Jeroen, and great moderation, Bert-Jan, and also for the very active discussion and all these questions. And thank you everyone for being here.